ELISA testing, a summary. What is ELISA testing? ELISA stands for Enzyme-Linked Immunosorbent Assay. It's a type of analytical laboratory test used to detect the presence of antibodies linked to a specific antigen. There are 4 types: direct, indirect, sandwich, and competitive. For this scenario, we'll only be focusing on indirect ELISA. ELISA serologies are usually performed in multi-well microtiter plates. The first step is having the antigens of the chosen pathogen immobilized on the well plate by binding to the walls. Antigens are the proteins or polysaccharides on the outside of a virus that trigger the immune response. Then the serum with primary antibodies are added to the well walls. Only the specific corresponding antibodies will bind to the antigens on the well walls. Antibodies in the serum not specific to the antigens will remain unbound. Then, the wells are rinsed to remove any unbound or incompatible antibodies. Then, a secondary enzyme-linked antibody is added to the wells. These proteins first bind to sites on the primary antibody. This secondary antibody is covalently conjugated to an enzyme. The wells are then rinsed again to remove excess secondary enzyme-linked antibody. Finally, a colorless substrate is added to the wells. It binds to the active site of the enzyme on the secondary antibody. This reaction catalyzes a visible color change. This is significant because it proves the existence of the relevant antibodies in the tested serum. So how can ELISA testing be applied? Well, in a local college dorm, there was an outbreak of an unknown disease affecting Sue Smith. Medical professionals analyzed a sample of Sue's saliva and isolated the pathogen DNA from hers. Through PCR cycle sequencing, the sequence of the pathogen's nucleotides or genetic code was discovered and analyzed with BLAST. It was determined with 97% accuracy that the disease was bacterial meningitis. However, it's not 100% certain. Therefore, ELISA testing must be performed with a serum sample to determine if it's bacterial meningitis. Following addition of a substrate, a color change would prove the existence of bacterial meningitis.